Today we are going to learn about sulfur amino acid metabolism. The first amino acid in sulfur amino acid group, that is methionine. You can see the sulfur in the structure of an amino acid. It is between two methyl groups which is present in a thiosulfur linkage. It is an essential amino acid and glucogenic metabolically. Degradation of methionine is result in the synthesis of cysteine, that is our second sulfur containing amino acid. Though cysteine is synthesized from the methionine, this methionine has a sparing action in the cysteine. That's why if we take methionine in our diet, so there is a less requirement of cysteine in diet because it can be synthesized from the methionine. Cysteine, it becomes non-essential because we can synthesize from the methionine, and obviously it is also a glucogenic amino acid. As we know, the cysteine is present in large quantity in the keratin of hairs and nails. Now let's see the metabolism of methionine. So this is a methionine amino acid. In the first step, this methionine is react with the ATP, that's adenosine triphosphate, and with the help of methionine adenosyl transferase, that means the transfer of adenosyl group to the methionine, resulting in formation of S-adenosyl methionine. This is an active form of methionine. SAM, that is SAM, S-adenosyl methionine. After activating methionine in the form of S-adenosyl methionine, it will become able to donate its methyl group. Then it acts as a methyl donor. So in the next reaction, any molecule that requires methyl will accept this methyl group from the S-adenosyl methionine, and that become methylated, and this SAM get converted into SAH, that is S-adenosyl homocysteine. S-adenosyl homocysteine. Now with the help of hydrolase enzyme, there is a release of adenosine, and the remaining molecule is called homocysteine. We will learn in detail about homocysteine later on, but this homocysteine is considered as a cholesterol of new era. In the next step, this homocysteine again get back converted to methionine by getting a methyl group, and that we get from the N5-methyl tetrahydrofolate. This methyl group transferred to the homocysteine and get converted into methionine, and here this tetrahydrofolate get converted into N5-methyl tetrahydrofolate get converted into tetrahydrofolate. For this reaction, what we require is vitamin B12 as a coenzyme. So this is a metabolism of methionine, in which methionine first is activated to S-adenosyl methionine, then it becomes a methyl donor. It donates its methyl group to any acceptor and gets converted into S-adenosyl homocysteine. This S-adenosyl homocysteine will releases the adenosine with the help of hydrolase enzyme and get converted into homocysteine. You will see the applied part of homocysteine in later part. This homocysteine accepts a methyl group from N5-methyl tetrahydrofolate, that is from folic acid, and get converted into methionine in the presence of vitamin B12. So folic acid and B12 is very essential for the metabolism of methionine. Now we'll see the various important roles of methionine. Once, as I told you, methionine is getting activated in the form of S-adenosyl methionine, then it plays important role in transmethylation reaction. It becomes a methyl donor. There are few important products that is synthesized with the help of transmethylation reaction from methionine, that is like synthesis of creatine, formation of N-methylbutyramide, epinephrine, metanephrine, normetanephrine, choline, anserine, melatonin. Again the choline from the serine, methylhistidine, lysine and methylated tRNA that are helpful for the translation process. So this is the various role of methionine by transmethylation reaction. We'll see few of them in detail. So the important product derived from methionine, the first one is creatine. We have seen in the previous video the glycine metabolism that when creatine is synthesized from the glycine requires three amino acids: glycine, arginine, and methionine. Methionine in the form of S-adenosyl methionine. It donates its methyl group to the guanidoacetic acid and forms a creatine. Itself gets converted into homocysteine. So the first thing that is synthesized from the methionine is creatine. Then epinephrine. We have seen this in the aromatic amino acid metabolism. When we have epinephrine, gets a methyl group from the S-adenosyl methionine here. Here you can see this is methyl group. This epinephrine getting converted into epinephrine. Then the choline. Choline from the glycine, serine, then ethanolamine, and after that it gets a methyl group one by one. And it gets converted into methyl ethanolamine, dimethyl ethanolamine, and trimethyl ethanolamine, and ultimately it gets converted into choline. That helps in the synthesis of acetylcholine. It's a methyl group that comes from the S-adenosyl methionine by the transmethylation reaction. Then the melatonin, again we have seen this in tryptophan metabolism. This 5-hydroxytryptamine, that is serotonin, getting converted into acetyl serotonin. And then this acetyl serotonin gets a methyl group that from the S-adenosyl methionine and get converted into melatonin. So this is again a transmethyl reaction that will be synthesis of one more neurotransmitter, that is melatonin. Now cysteine synthesis. In our previous slide, we have seen that the methionine get converted into SAM, that is S-adenosyl methionine. After donating, methyl group get converted into SAH, that is S-adenosyl homocysteine. And after removal of adenosyl group, it gets converted into homocysteine. Now this homocysteine react with the serine, that is a hydroxyl group containing amino acid, and in the presence of cystathionine synthase enzyme, it forms a cystathionine. For this reaction, what we required is pyridoxal phosphate as a coenzyme, PLP. So here there is the role of vitamin B6 in the metabolism of methionine and cysteine. After that, the cystathionine acted upon by cystathioninase enzyme and splits the cystathionine to cysteine and homoserine, and that homoserine get converted into ultimately propionyl CoA, and we know this propionyl CoA gets converted into succinyl CoA by the successive reaction, and this succinyl CoA enters into the TCA cycle as an intermediate, and it forms an oxaloacetate, and that oxaloacetate to the gluconeogenesis, and it synthesizes the glucose. So hence methionine or cysteine is glucogenic. So here there is a summarization of conversion of methionine to cysteine. As I told you in the beginning, methionine converted into S-adenosyl methionine will get converted into S-adenosyl homocysteine. After removal of adenosine group, it get converted into homocysteine. If there is a B12 and methyl tetrahydrofolate, it get back to the methionine, or this homocysteine react with the serine. In the presence of B6, forms a cystathionine, and again it splits to cysteine and homoserine. So here is the conversion of methionine to cysteine. For this, what we require is three important water-soluble vitamins, that is vitamin B6, vitamin B12, and folic acid. Now, the degradation of cysteine. What are the different end products we get from the cysteine. From the cysteine, by the transamination reaction, we get beta mercaptopyruvate, and then after that we get the pyruvate. And you know, this pyruvate enters into the gluconeogenesis pathway. Second, formation of beta mercaptoethanolamine, that is essential for the synthesis of coenzyme A, cysteine. By the decarboxylation reaction, forms beta mercaptoethanolamine, and this beta mercaptoethanolamine is essential for the coenzyme A synthesis. The sulfur present in the cysteine may be removed either as a H2S, the hydrogen sulfide, or elemental sulfur, or as a sulfide, and the remaining carbon skeleton utilized for the synthesis of other products. Now we'll see the various metabolic functions of cysteine. As we have already seen in the glycine metabolism, there's three amino acids, glycine, glutamate, and cysteine, that is essential for the synthesis of glutathione. So it forms a gamma glutamate cysteine glycine, that is glutathione. It has a various role in the various metabolic pathways. The first important role of glutathione is amino acid transport. So this glutathione comes from the cysteine, and that helps in the amino acid absorption by the Mester's cycle. This glutathione can easily be reduced and oxidized, so it's helpful in the redox reactions and play a very important coenzyme role along with the oxidoreductase group of enzymes. The metabolic role of reduced glutathione is mainly in the reduction reaction, where it releases the hydrogen that is accepted by the substrate. RBC membrane integrity. Again, it is a very essential role of glutathione, but synthesized from the cysteine, plays very essential in maintaining the RBC membrane integrity. In RBC, now free radicals are generated, and the removal of this free radical is very important. If it is not removed, that can damage the red blood cell membrane and it leads to hemolysis. These free radicals get converted into hydrogen peroxide with the help of superoxide dismutase. Glutathione peroxidase that convert this hydrogen peroxide to H2O. For that, this glutathione peroxidase to act, what we required is reduced glutathione. So this reduced glutathione helps glutathione peroxidase to convert this hydrogen peroxide to water. And this reduced glutathione regenerated with the help of glutathione reductase by using NADPH that comes from the HMP shunt pathway in red blood cell membrane. Then activation of enzymes. Other functions of cysteine is a conjugation for detoxification reaction. Detoxification means conversion of waste metabolites from water insoluble to soluble compound by the process of conjugation, so that we can excrete that out. Glutathione helps in the detoxification of several compounds by transferring its cystinyl group, for example, organophosphorus compound, halogenated compounds, nitrogenous substances, other heavy metals, and the drugs. So this glutathione reacts with this compound, that is R suggestive of any compound, will take the cystinyl group that forms an acetyl cysteine product, and that will be excreted in urine. Second function, conversion of methemoglobin to hemoglobin. Methemoglobin iron is present in form of Fe plus 3, and that gets converted into hemoglobin with the help of glutathione. Formation of taurine. Cysteine converted into cystic acid and by the decarboxylation get converted into taurine. And this taurine helps in the formation of by source, that is taurocolor, keeping the correct structure of protein as a cysteine present in the structure of protein helpful in the formation of interchain or intrachain disulfide groups, and it is very essential to keep the structure of protein in the correct dimension. Now here there is a summarization of methionine and cysteine metabolism. Methionine activated into S-adenosyl methionine that helps in the transmethylation reaction. By donating the methyl, it helps in the synthesis of various important compounds like creatine, epinephrine, choline, melatonin, and so on. After that, this SAM gets converted into S-adenosyl homocysteine, and that forms a homocysteine that reacts with the serine and helps in the synthesis of other sulfur group containing amino acid, that is cysteine. Being a glucogenic, it converted into pyruvate that forms a glucose. The cysteine helps in the formation of coenzyme A, taurine, and essential for the glutathione synthesis. So this is all about the metabolism of methionine and cysteine. Now we go through the applied part related with the metabolism of sulfur amino acid metabolism. There are few inborn errors of sulfur containing amino acid metabolism. One of them is cystinuria. Name itself suggests excretion of cysteine in urine. It is an autosomal recessive condition, and it is one of the inborn error of Gerrard-Stetter. It is mainly due to defect in amino acid transport. Sign symptoms that includes precipitation of renal cysteine as a calculi in the renal tubules, abnormal excretion of cysteine and lysine in the urine, and in acidic pH, the cysteine crystals formed in the urine that is excreted and forms of cystinuria. This cystinuria diagnosed with the help of cyanide nitroprusside test. It is a screening test, and for the treatment, the drug of choice is penicillamine, and the other ways by increasing the urinary output by increasing the fluid intake or making the urine alkaline by the various alkalizers. Second defect, that is cystinosis. Again, it is autosomal recessive condition due to abnormality in transport of cysteine, resulting in deposition of cysteine crystals in various tissues in the organs, that is accumulated in liver, spleen, bone marrow, kidney, cornea, lymph nodes, and the white blood cells. It resulting in generalized amino aciduria, and the renal functions are also affected in this stenosis. Treatment: give adequate fluid intake, so increase the urine output for washing out of this amino acid. Make urine alkaline by using sodium bicarbonate, and the drug of choice is penicillamine for the cystinosis. Another inborn error for the sulfur-containing amino acid metabolism is homocystinuria. It is a autosomal recessive condition. The incidence rate is 1 in 2 lakh live births. The normal homocysteine level in blood is 5 to 15 micromoles per liter, but it may increase in certain diseases to 50 to 100 times. The moderate increase in aged person, so it's increasing along with the age. It also increases in B12 and B6 deficiency, because we have seen in the metabolism this B12 and B6 is essential for the synthesis of this homocysteine to either methionine or to the cysteine. It also increases in the tobacco smokers, in alcoholics, and in the patient with the hypothyroidism. There are mainly two types of homocystinuria. One is typical homocystinuria type 1, and the second is cystinuria. We'll see one by one. Homocystinuria type 1, that is mainly due to defect in cystathionine synthase enzyme. The cystathionine synthase converts homocysteine and serine to cystathionine. There will be an abnormal accumulation of homocysteine and methionine. Signs and symptoms include mental retardation, Charlie Chaplin gait, skeletal deformity, ectopia lentis, myopia, and the glaucoma. This elevated homocysteine activates the Hagman factors of coagulation pathway, causing increased platelet aggregation and resulting in thrombosis. Because of that, this homocysteine has a higher level of homocysteine associated with the cardiovascular diseases. This condition can be diagnosed with type of cyanoid nitroprusside test that will be positive in case of homocystinuria. Treatment includes diet give low in methionine because if excess methionine gets converted into homocysteine and that will be accumulated. Use diet rich in cysteine because of the deficiency of this enzyme or the defect in this enzyme, methionine cannot get converted into cysteine. Along with that, there will be increased supplementation of vitamin B6 is required. Second condition, that is cystinuria. It is due to defect in cystathionase enzyme that converts cystathionine to the cysteine and homocell. It is an autosomal recessive condition. It is also associated with the similar symptoms like of typical homocystinuria type 1 like mental retardation, anemia, thrombocytopenia, involves eye, that is ectopia lentis, myopia, glaucoma, along with the other endocrinopathies. B6 deficiency may sometimes associate with the cystinuria because the cystathioninase enzyme required vitamin B6 for its activity. Diagnosis in this cyanide nitroprusside test is negative, but the cystathionine can be detected in the urine. Treatment again the same, gives diet with the low methionine and the high cysteine. And the supplementation of B6, folic acid, and B12, that is copalamin, is essential for the treatment of cystine metabolism. So this is the summarization of inborn aerobic sulfur containing amino acid metabolism, cystinuria, homocystinuria, and cystinuria. Cystinuria is mainly due to defect in transport system. This homocystinuria is due to defect in cystathionine synthesis, there is accumulation of homocysteine. And cystathionine is due to defect in cystathioninase, resulting in increased cystathionine in the blood. These conditions both associated with the mental retardation mainly. In homocystinuria, ectopia lentis is a prominent one. In cystinuria, renal calculus is seen. Excretion of cysteine in the cystinuria, homocystinuria, and cystathionine. The cystinuria and homocystinuria gives cyanide nitroprusside test positive, while the cystathionuria gives negative. Supplements, fluid, and alkalization of urine is essential for the cystinuria. While in homocystinuria and again in the cystathionuria, cysteine in diet is essential, methionine is restricted in the diet, and B6, B12 and the folic acid supplementation has to be given in these two conditions. Now, in the beginning, as I told you, this homocysteine is considered as a cholesterol of new era because we know the hypercholesterolemia leads to cardiac disease. The same way, there is a relation of homocysteine and heart disease. But as I told you, the normal level of homocysteine is 5 to 15 micromole per liter. But increase of 5 micromole per liter of homocysteine is equivalent to increased cholesterol up to 20 milligram per deciliter. So it also increases the risk of coronary artery disease that increase in the homocysteine level. This elevated homocysteine interacts with the lysyl residues of collagen and increase the tendency for atherogenesis. And atherogenesis affects the and leads to various coronary artery diseases. For this, the supplementation of this pyridoxine vitamin B12, that is cyanocobalamin, and folic acid. It's very helpful to keep this homocysteine level within the normal level. So this is all about metabolism of sulfur containing amino acid, that is methionine and cysteine. We have seen detailed metabolism of cysteine, methionine, what is the important role of methionine and cysteine, and associated inborn error of metabolism. Thank you. For more updates, you can visit Insta account, that is @biochemistry for student, and you can visit the same YouTube channel, Biochemistry for Student, for other important topics.